All right, this is the last video of the Tuesday for week 6 lecture. So we just talked about transcription, which is the production of functional RNA molecules. And then translation is going to use those RNA molecules. It's going to use the messenger RNA as the template for actually synthesizing a polypeptide. It's going to use the tRNA as a delivery system for the amino acids for that process of polypeptide synthesis, and then it's going to use the ribosomal RNA as the actual active site and platform on which the polypeptide synthesis is going to occur. So again, there's going to be initiation, elongation, and termination. This is a super important process in cells. This is where actually a large part of the energy of cells goes. If you look at this quote, that protein biosynthesis is by far the largest consumer of energy during cellular proliferation. So that is basically growth of cells. Translation by ribosomes is estimated to account for half of the energy consumption in a rapidly growing bacterial cell, and the majority of the ATP is going in one way or another into that process. All right. During the process of translation, the ribosomes interact with the messenger RNA. So if we have this diagram here, this is the messenger RNA down here, these are the tRNAs down here. Essentially all around this, the context in which this is all going to happen is going to be on the ribosome, right? So the tRNAs are going to bring the new amino acids in to form peptide bonds with the growing chain. The growing chain is sort of transiently attached to the tRNA still, right? So this one is going to attach to this one, and now this one's going to have the growing polypeptide chain on it, right? These are the codons down here. Remember, a codon is 3 nucleotides in the RNA sequence, right? 3 successive nucleotides. And then the peptide bonds are going to be formed up here, okay? So prior to starting translation, each tRNA has to actually be charged with the right amino acid, right? So that process is done by enzymes called aminoacyl tRNA synthetases, right? So this purple blob here is the tRNA synthetase. So it's going to recognize a very specific tRNA molecule, and it's going to put on it a very specific amino acid, right? So there are different aminoacyl tRNA synthetases for each amino acid, right? And so they're going to recognize the tRNAs, they're going to recognize the amino acid, and they're going to attach them together. This is critical for the fidelity of the translation process, so that when this codon, this is the anti-codon on the tRNA, but it's going to partner with a codon on the messenger RNA, and that's going to put the right amino acid onto the growing peptide chain. Okay, so again, video here. I want you to watch this so you can actually see it in action, because the way I'm describing it is not going to be sufficient. So please watch these videos so you can see sort of translation in action, right? So translation is going to depend on the interaction between the ribosome subunits, in particular the 16S ribosomal RNA, and a sequence on the messenger RNA called the Shine-Dalgarno sequence. This is basically upstream of the start codon in a bacterial messenger RNA, right? So this is sort of positioning the ribosome or the messenger RNA on the ribosome so that the start codon, which is a methionine codon, is going to be in the right position on the ribosome. So most, not all, but most polypeptides are going to start with a methionine codon, this AUG codon, right? So we also refer to this as the start codon. This Shine-Dalgarno sequence, I like to refer to it also as the ribosome binding site, because that's a little more descriptive. Shine and Dalgarno are the names of the scientists that discovered it, but ribosome binding site tells you what it's doing. It's positioning the messenger RNA on the ribosome so that the start codon will be in the right place. Okay, this is just showing the composition of eukaryotic and bacterial RNAs, including the various protein components and the ribosomal RNA components. Again, the 16S ribosomal RNA is really important for actually recognizing this ribosome binding site. It's actually complementary to a sequence in the 16S ribosomal RNA. Okay, this is actually the last slide, and it just makes a point on bacterial specific translation, okay? And here's the point. A bacterial messenger RNA can actually have multiple coding regions on it, okay? So this, you could think of this as, for example, the Lac Operon Messenger RNA. If you remember back to what you learned about the Lac Operon, it has 3 genes in it, Z, Y, and A, okay? Lac Z, Lac Y, and Lac A. So this is just generically gene A, gene B, gene C. A bacterial transcript does not have to have specifically 3 genes. It could have 1, it could have 10. The point is that it can have multiple, okay? So the reason that it can have multiple coding regions versus a eukaryotic messenger RNA, which does not have multiple coding regions, is that each start site, each start codon, which is a methionine codon, has a site that tells the ribosome that this is a start codon. Remember, methionine is just one of the 20 amino acids, and it can be found other places, not just as the start codon for a polypeptide, right? So there could be an AUG here, an AUG here, an AUG here, etc. There can be multiple methionine codons. So the bacterial ribosome needs to know which one is actually the start codon if you're going to have multiple genes in there. So this ribosome binding site or Shine-Dalgarno sequence tells it, hey, the methionine codon right next to this is where you should start. Now, a eukaryotic transcript doesn't have this separate ribosome binding site. So the ribosome is just going to start at the end and go until it gets to the first methionine codon, and that's going to be the start of translation. That's the only place translation is going to start, right? So if there was another gene down here, it would never get translated, okay? But bacteria can avoid that by telling the ribosome, this is a start codon, okay? That means you can have multiple, as I said, you can have multiple coding regions on a single transcript. We call that a polycystronic messenger RNA. Cistron is just an old name for a gene, okay? So eukaryotic messenger RNAs are monocistronic. They only have 1 translated gene on them, but bacteria can have several, okay? A set of genes that is all transcribed from the same promoter. So this is a 5' end of the messenger RNA, and that's going to be where the RNA polymerase started, okay? So that's going to be positioned next to the promoter on the DNA, right? So the DNA here would look like promoter here, gene number 1, gene number 2, gene number 3, and then terminator down here, okay? I'll draw that on the board in class, so the recording from the class will show that as well, okay? But the point is there's just 1 promoter and 1 terminator, and all the genes in between are going to be transcribed as part of the same messenger RNA, okay? We call genes that are organized so that they're all transcribed on 1 messenger RNA an operon, okay? So O-P-E-R-O-N, operon. That's important because the genes that are part of the same operon usually are all regulated at the same promoter, and they're usually functionally related to each other. So they may be involved in the same metabolic pathway. They may all be involved in making the same structure, like a flagellum or a lipopolysaccharide or something like that. So the genes that are part of an operon are functionally related to each other, and they are co-regulated, okay? And in bacteria, the fact that these can be linked together on the same transcript facilitates that co-regulation. The other important thing that's shown here is that the the ribosome can be translating all of these simultaneously. And although you can't see it here, it's also important to remember that because there's no nuclear membrane in a bacterial cell, the ribosome can actually be working on this first gene while the messenger RNA is still being made down here. So this is showing like a fully synthesized messenger RNA, but in fact, you can have translation happening while transcription is still occurring. Okay, they can be functional, they can be spatially connected to each other, right? So remember, the nucleoid in a bacterial cell does not have a membrane around it, so the ribosome can access that messenger RNA while it's still coming off the DNA. Okay, so I'm going to end it there. Let's see, I'm going to stop sharing, and I'm also going to stop recording here. Okay, this is the last one.